Polymerase chain reaction. My part in this presentation will be to look at the purpose, what it is used for, and also go into the history and development of this biological technique. Well, firstly, what exactly does polymerase chain reaction mean? Using a dictionary, polymerase is defined as an enzyme which brings about the formation of a particular polymer, especially DNA or RNA. Chain is defined as a sequence of items of the same type forming a line, and reaction is defined as a chemical process in which substances act mutually on each other and are changed into different substances, or one substance changes into another substance. But what does this all mean? As a combined term, polymerase chain reaction can be defined as a technique used to amplify or to make many copies of a specific target region of DNA. Purpose. Now that we have defined PCR, we can derive a purpose from the meaning. So ultimately, the purpose of polymerase chain reaction is to make copies of DNA sequences in large quantities. This was a major breakthrough in molecular biology and is extremely purposeful in many ways across the scientific community. Figure 1 here shows just how DNA polymerizes and then the chain reaction occurs, making multiple copies. In a real life scenario though, there would be billions amplified. This is not a small scale technique. This technique has allowed many advancements and great benefits in medical fields in areas of microbiology, dentistry, parasitology, and virology. Within these fields, it can detect problems such as bacterial, fungal, or viral pathogens, and also identify any disease strains found in genes. PCR has the ability to prenatally diagnose hereditary diseases and also distinguish the expression and structure of genes. But not only does PCR have benefits in medical fields, it also services areas of forensics, in scenarios of DNA typing, genetic fingerprinting, and also DNA testing. Archaeological analysis has discovered an entirely new field from the use of PCR, from the efficacy of its ability to produce billions of DNA copies from simply just one sample. History and development. PCR was developed from an amalgamation of previous discoveries relating to DNA. So just diving into the history, we are going to look back to all the discoveries throughout the years that led to the discovery of polymerase chain reaction. So first of all, Watson and Crick in 1953 identified the structure of DNA to be a double helix, revealing paired bases running along two strands in opposing directions. In 1957, Arthur Kornberg identified DNA polymerase for the very first time. Gobind-Khorana in 1971 played a part in the discovery of the genetic code. This led to research in simplifying genes using primers that were artificial and templates that assisted in DNA polymerase to replicate specific gene segments. Frederick Sanger in 1977 discovered the final components needed to create PCR. He recognized a method of DNA sequencing that required a DNA polymerase, nucleotide precursors, and a primer. So now all the factors for PCR amplification were available. Finally, in 1983, Kary Mullis discovered polymerase chain reaction. Dr. Kary Banks Mullis, a chemist who won the Nobel Prize in 1993 for the discovery of polymerase chain reaction. He worked at Cedars Corporation doing research about oligonucleotides when he was faced with a few problems. Unconsciously, by combining his issues, he began devising an array of methods that related to oligonucleotides, DNA extension, and DNA polymerase. The result was the invention of polymerase chain reaction.
Polymerase chain reaction is an enzymatic assay that amplifies a small amount of a specific DNA fragment from a larger pool of DNA, which was developed in the late 1980s. This process makes billions of copies of the specific DNA fragment that enables detection and subsequent identification of gene sequences using visualization techniques based on size, that is molecular mass, and charge. There are 4 key ingredients involved in polymerase chain reaction, that is, template DNA, primers, nucleotides, and DNA polymerase. Template DNA is the genetic material obtained from a variety of tissues and organisms that are to be investigated. It is generally double-stranded DNA. The source DNA is extracted from hair, blood, skin, saliva, and microbes. Only small trace amounts of DNA are required for PCR to be performed, making it useful in the study of specimens which only have a small amount of viable DNA. RNA that is converted to cDNA can also be used as well for the basic template, which is referred to as reverse transcription PCR. The primers are short DNA fragments called oligonucleotides that are usually 18 to 22 bases in length, with a sequence complementary to the target DNA that will be detected and amplified. Primers in the polymerase chain reaction pinpoint the exact DNA product that will be amplified. Primers serve as an extension point for DNA polymerase to build upon. Nucleotides act as building blocks that are used by the DNA polymerase to create the PCR product. These include adenine and guanine, which are purines, and cytosine and thymine, which are pyrimidines. DNA polymerase is the key enzyme which joins individual free nucleotides together to form the PCR DNA product. Initially, in developmental stages of PCR, DNA polymerase from E. coli was used, but had to be constantly added during each cycle of the PCR process, which was known as the Clino fragment. Thermophilic bacteria, Thermus aquaticus, contains Taq polymerase enzyme that is stable at high denaturing temperatures, which replace the DNA polymerase from E. coli, as this issue could be overcome. The PCR reaction consists of 3 major steps that are denaturation, annealing, and extension. The first step is denaturation. Initially, the reaction chamber is heated to a high temperature of around 95 degrees Celsius for a period of 20 to 30 seconds. This causes the double-stranded DNA template to denature via the breaking of hydrogen bonds between complementary base pairs, adenine and thymine, and cytosine and guanine, that gives 2 single-stranded DNA molecules. The second step of the process is annealing. The temperature is then lowered to 68 degrees Celsius for about 20 to 40 seconds. That allows the specific oligonucleotide primers to bind to target DNA segments. This process is annealing. Annealing between target DNA and oligonucleotides occurs under the condition that they are complementary in sequence to each other. Temperature is low enough to allow for hybridization of primer to strand, but sufficient for hybridization to be specific. The third step of the process is elongation. The temperature is raised to 72 degrees Celsius for 20 to 40 seconds so that the thermostable Taq polymerase synthesizes a new DNA strand complementary to DNA template strand. In the 5 to 3' direction, free deoxyribonucleoside triphosphates, DNTPS, are added, resulting in the condensation of 5' phosphate group of DNTPS with 3' hydroxy group at the end of the elongating DNA strand. This process results in selective enrichment of specific DNA sequences that can be readily picked up and modified. The entire process is then repeated 20 to 60 times, and an amplification factor of 1 quadrillion can be attained. Current applications of PCR include DNA fingerprinting, where PCR is employed to identify or exclude subjects of interest by analysis of small amounts of blood, semen, or other biological material left in the crime scene. It is also used in the detection of pathogens, where PCR is able to easily detect as little as 10 integrated copies of HIV in the presence of 100,000 uninfected lymphocytes in a range of biological contexts, such as tissue culture cells, seronegative patients, hemophiliacs, and AIDS patients. PCR is also a useful tool in the detection of diseases, such as hemophilia B, where there is a mutation for the coagulation factor 9 that alters the size of diagnostic restriction fragments by increasing the amount of this fragment. Another current application of PCR includes the detection of dental caries in dentistry. In dental caries, 7 species of cariogenic bacteria are prevalent in the oral cavity that include Streptococcus crescentus, rati, mutans, Sobrinus, Downae, Ferus, and Macacae. To ensure a rapid, simple, and sensitive procedure for the species identification and differentiation in the oral cavity, PCR is used in place of various biochemical, immunological, and genetic methods. PCR proves useful in identifying species associated with dental caries and sites in ecological niches, thus enabling a better understanding on the process of carious developments in the dentition. Discriminative power of PCR between species provides a comprehensive way of determining caries risk in patients and monitoring the efficacy of therapeutic and preventative interventions. Another current application of PCR is in virology. The detection of viral nucleic acids and their characterization has been made possible by real-time PCR. Real-time PCR is useful in the quantification of more diverse sequences of viral nucleic acids of infection-causing viruses in a single reaction. It's particularly valued for its rapid nature in detection of viral variants and syndromes caused by such virological pathologies. A potential future application of PCR includes qualitative detection of rare fetal specific events. Digital PCR, due to its high sensitivity to develop adapted assays, makes it a lucrative qualitative detection option. Celence's 2015 work that demonstrated single-copy fetal DNA sequence targets to be all detected by digital PCR compared with real-time PCR highlights its potential use for improved accuracy for fetal sex determination and RHD genotyping, especially for suboptimal samples with low fetal DNA proportions of 2%. Another potential future application is the investigation of differentially methylated loci in different organs, such as the placenta. The TIMP3 promoter region that is hypomethylated in DNA from preeclampsia placenta relative to the DNA from maternal blood has reported to be a potential prenatal diagnosis marker for preeclampsia in the maternal plasma. Digital PCR has been able to quantify differentially methylated loci after a pre-analytical step of methylation-sensitive digestion or bisulfite treatment. This serves as an effective method for the diagnosis and monitoring of pregnancy-associated disorders like preeclampsia.
PCR, like any technique, has advantages and limitations. These advantages and limitations should be considered when deciding if this technique is right for the application. Over the following slides, we will discuss this in further detail. PCR is a very sensitive technique. Using PCR, DNA can be amplified from tiny amounts of the sample. Just one cell is needed to create millions of copies. This makes it useful for situations such as DNA fingerprinting, where only a small amount of trace DNA may be present, but the sample needs to be amplified in order to be analyzed. Additionally, this small amount makes it a good technique for amplifying samples that may be old or degraded, making it useful in applications like archaeology. Another advantage of PCR is it is reasonably simple to understand, both in performing and in concept. This makes it a technique that is accessible to a wider range of skill sets, rather than the users of the technique having to be overly specialized. Due to the way PCR replicates DNA, results are available very quickly. This makes PCR a great technique when results are time sensitive. An example of this is detecting if someone has a certain illness in which treatment is more effective the sooner it is commenced. This advantage also brings about the ability to speed up the time it takes to do and repeat experiments. Unlike some techniques that could take days, PCR allows experimenters to get results fast, and if errors were made in experiments, they can be repeated without considerable loss of time. Now that we have looked at some of the advantages of PCR, we are going to move on to some of the limitations. The first one we're going to look at is that some prior information is needed. In order to create DNA primers, the experimenter needs to know some information about the DNA they are looking to amplify. As they must create primers for each end of the sequence, they need to already know the sequence information at the regions they are going to be using. This does limit PCR to applications where we already know information about the material we're using or looking for. Another limitation of PCR is that DNA polymerases are prone to error. These errors can lead to mutation in the product that is generated. Experiments such as cloning require a high fidelity result, that is, the result that is identical to the original DNA, with little to no errors. Errors in duplication could lead to unusable or misleading results. Some DNA polymerases are less prone to errors than others, and this information can be used to help mitigate this particular limitation of PCR. As stated previously as an advantage, the fact that PCR is a sensitive technique is also one of its shortcomings. Even the tiniest amount of contamination in the sample could lead to misleading results. An example of this is explored in an article about using PCR to characterize transgenic plants. Wassinger explains that a limitation in PCR they come across regularly was the fact that some plants would have traces of agrobacteria surviving. This agrobacteria would then get amplified in place of the plant sample they were looking to explore, resulting in a very misleading result. Thank you so much for listening to our presentation.